Very good evening, everyone. Please confirm if you all can hear me. Hello, Fadia. The session has started. I hope you can hear me and others. Okay. Thank you for confirming. So, welcome all the Biotecnika and Rasayanika students, our subscribers, and everyone who has joined us for this webinar. Have you ever wondered how breakthrough discoveries in pharmaceuticals, food safety, or environmental science happen so precisely and reliably? Well, the answer often lies in advanced analytical techniques like gas chromatography, liquid chromatography, and mass spectrometry. Hello, everyone. I'm your host, Dr. Tanushree, and I'm excited to have you all join us for this power packed webinar designed specially for you. Whether you are a research aspirant, analytical chemist, or life science enthusiast who are eager to understand the practical applications and career potential of GC/LC/MS technologies, then you are at the right place. In today's webinar, you will explore why mastering GC/LC/MS techniques is not optional, but a must-have skill for every research aspirant and analytical chemist. So, to enlighten us on this today, we have with us two very experienced and eminent personalities. Let me introduce our esteemed speakers for today. We have with us Mr. Mohammad Khaja Mohinuddin, analytical scientist from Dr. Reddy's Laboratories. Mr. Mohinuddin is a passionate and research-oriented analytical scientist with over 13 years of extensive experience in the pharmaceutical industry. He specializes in method development and validation for drug product impurity profiling, including genotoxic impurity analysis using advanced techniques such as LCMS and GCMS. Mr. Mohinuddin is highly skilled in analytical techniques, including HPLC, GC, mass spectrometry, TGA, DSC, PXRD, and particle size analysis. He has significant expertise in developing and validating distribution methods using USP apparatus and is well versed in working under CGMP compliant environments. Mr. Mohinuddin in his session will share insights on career paths, industry opportunities, certifications, and skills required to strategically build your career in the field of analytical chemistry. Also, we have with us Dr. Anirudh Sharma, who is an accomplished chemistry educator and researcher with expertise in mass spectrometry and molecular modeling. As academic support specialist at Biotecnika, he develops and delivers interactive mass spectrometry courses, making complex topic accessible for students. He holds a PhD in analytical chemistry from Teesside University, London, and MSc in organic chemistry from VIT Vellore, and a BSc Honours in chemistry from University of Delhi. Dr. Anirudh's career includes academic roles in India and the UK. He's an acclaimed research publications and numerous honors, including Teesside University Doctoral Scholarship and Society's Personality of the Year by Teesside University Student Union. Dr. Anirudh, in his session, will take us through the fascinating world of GC/LC/MS, starting with the fundamentals and its powerful role in driving innovations across industries and research. Sir, we are truly honored to have you both with us today.
Thank you so much, Dr. Tanushree, for your warm welcome, and I'm looking forward to this session.
Thank you. Thank you so much, sir. So, everybody, before we dive in, a quick note. Please feel free to post your questions in the chat box. We will address them during interactive Q&A session towards the end. Also, we have a special surprise gift for you all. So stay connected with us. We will share it anytime during the webinar. So let's begin with our first session, and I would like to invite Dr. Anirudh sir. So over to you, sir.
Thank you, Tanushree, for such a warm welcome. I'm much grateful. Can I just check, are my slides visible?
Yes, sir, it's visible. Yeah, it's absolutely perfect.
All right. Okay. Thank you again, Tanushree. And hello, everyone, wherever you're joining from. I hope you're from India. So, good evening. Uh, Very warm welcome to the Biotecnika and Rasayanika students. So, for today, our agenda is we are going to talk about in the realm of gas chromatography, liquid chromatography, and mass spectrometry. We'll try and understand what it is. We'll do an introduction. Then we'll focus on why is GCLC and MS important. What are the real world application? From there on, we'll directly focus on you. That why research aspirant, all analytical chemists like you should focus on mastering these techniques. Then we'll discuss few case studies where GCLCMS have been a driving factor and has changed the how the world sees chemistry or biology. Then we'll talk about current global trends of what is happening in the world of GCLC and around the globe, and what is the future outlook for this analytical technique. And we'll conclude with the growing demand of analytical scientists who have expertise. As this poster for this webinar said, 70% of entry-level roles in the pharma industry needs a GCLCMS as one of their prerequisite. So we'll end the session on that, and I'll be more than happy to take your questions as well. So let's go exactly to the very beginning. That what exactly is chromatography? At its heart, it's simply a powerful way to separate mixtures. Think about it like this: you have a group of friends and you all decide to run a race. Remember the tortoise and the rabbit race you used to listen in the kindergarten? Now, in the race, some of you might be faster, while others like to stop and chat along the way, while others might get distracted by the interesting things and the scenery of the path. In chromatography, our runners are our molecules in our sample. The track they are running on is normally said to be the stationary phase, and the wind or current that carries them is the mobile phase. Different molecules interact differently with these two phases, causing them to separate over time. Our ultimate goal is to successfully separate each component of our mixture so we can identify what it is, how much of it is there, or even purify it. So, the main aim of chromatography is three. The first is to identify what it is. What is the identity of my sample? What are the components? If it is a pharmaceutical sample, it should ideally have a API and some excipients, and the scope goes on. It also helps us quantify how much of each component exists in the sample, and we can quantify. And again, the third main goal is to even purify it. We can take the samples in different vials and we can purify our mixture into its pure components. Gas chromatography, often abbreviated GC, is a foundational analytical tool for separating and analyzing compounds that can be vaporized without decomposition. It majorly takes place all in the gas phase. It is essential in industries from environmental monitoring to pharmaceuticals, and there is no analytical lab which exists without gas chromatography. Liquid chromatography, on the other hand, is a technique used for separating and identifying molecules which are dissolved in a liquid. So in gas chromatography, we have the sorry, apologies about that. In gas chromatography, we have the gas as the main phase, but in liquid chromatography, our sample is typically dissolved in a liquid and hence it is analyzed. It is indispensable in analytical testing labs. There is no, again, I would like to emphasize, no analytical technique exists, no analytical lab exists without chromatography, be it GC or LC. I'm sure Mr. Khaza will agree with me because he works on pharmaceuticals the entire day. Now, I have a very, very simple question that why should budding scientists, budding researchers like yourself should focus on learning GCLCMS? Why should we take the pain to do it? And the answer is very simple. This is because gas chromatography, liquid chromatography, and mass spectrometry are the workhorses in pharmaceutical development, environmental monitoring, forensic investigation, and food safety. This pretty much, combines all the 70 to 80% things you come across in your daily life. Mastering these tools means you are able to answer real world questions that matter. Think about the paracetamol or the ibuprofen that you take when you have a fever or you have pain. This that medicine has gone through a rigorous process of testing, and that could not have been possible without GC, LC, and MS. So let's go to our next part of why separate. You might be asking, if mass spectrometry is so powerful for identification, which it is, why do we need to separate things first? Why should we bother about putting a GC to a mass spectrometer or a LC? Apologies, to a mass spectrometer. And if this question is in your mind, please feel free to write it in the chat box. I'll address all the questions in the end. And if you have this question, this is an excellent question. The reality is most real world samples, whether it's the blood sample, a plant extract, or a complex industrial chemical, all these are incredibly complex mixture. There are too much of different components in them. While mass spectrometry is brilliant at identifying individual molecules, it can get overwhelmed when too many different compounds hit it all at once, especially when they have similar masses. This can lead to what is known as the matrix effect, where one compound interfaces with the ionization of another. This is where chromatography becomes indispensable, and it is very important at this point. It meticulously separate these complex mixtures into individual components, or at least reduces their complexity significantly. This basically leads to two advantages before our sample can reach the mass spectrometer. This pre-separation enhances both the specificity, meaning we are truly measuring what we want to measure. If we want to measure caffeine, we are measuring caffeine and nothing else. And the second thing is it increases the sensitivity, which allows us to detect even tiny amounts of our analysis. In pharmaceutical, there are impurities always, and there is a certain regulation from the government of, for example, your impurities can be in the ppm level, parts per million, or ppb, parts per billion level. This is where hyphenation becomes very helpful. This is why we hyphenate these techniques: chromatography for separation and mass spectrometry for identification and quantification. Together, they create an incredibly powerful analytical tool. Now, let's understand what is the real world implication. Where does in the real world is GCLC and MS useful? The first is the pharmaceutical sector. As I said previously, all the medicines that you consume, be it a supplement, be it the allopathic medicine or anything similar, goes through a regular process of testing, which cannot be completed without this analytical technique. From drug development to impurity testing, and even understanding how these medicines work to their pharmacokinetics. These analytical techniques play a major role in pharmaceutical. The next is environmental. This global warming is on the top, and you must have seen the news that uh, microplastics are already in our body and there's no way it can go out. A very good analytical technique to identify these pesticides which come from the soil per polyfluoroaromatic systems, which is named as the forever chemicals, which don't go away, and microplastics in water or the soil. The second industry is food and beverages. This includes the soft drink, the food that you eat, consume, things from allergen testing to authenticity, to authentify if a company is stating that a certain amount of product has 40 milligram of caffeine, we can use this analytical technique to identify that this is true or not. And very importantly, in forensic. Have you ever imagined if someone who is accused of having drug, or maybe in a illicit drug, which is a recreational drug, which they're not supposed to have, how do they actually convict them in the court? Because in the law of court, we need evidence. It is these powerful analytical techniques which helps in the identification of opiates, which are recreational drug, arson investigation, where someone might have uh, put a building to fire, or so on. And lastly, the petrochemical industry. These analytical techniques, specifically GCLC and MS, are very important. They help in the petrochemical industry to estimate the fuel quality and to understand what is the hydrocarbon. Hydrocarbon means carbon and hydrogen component of a fuel sample. Our next aim is about the importance of what you can actually gain. Why is it importance? Why should we put our time and effort to mastering GCLC and MS? The very first rule is stricter regulations. Science has developed very rapidly in the last 3-4 decades, and the regulations from companies like the FDA, which is in the US, EPA, and EU, they require very tough and rigorous testing of all the pharmaceutical compounds. This is mainly to avoid things like the thalidomide incident, which leads to so many deaths. So the regulations are a lot stricter these days. The second is technological advancement. 40 years ago, doing high resolution mass spectrometry was a very big thing, or doing tandem mass spectrometry, where we do two stages of mass spectrometry for complex analysis, was very rare. But with the advent of new mass spectrometer with various companies like Waters, Perkin, and Thermo Fisher, it is now possible to do this routine analysis in a quicker and in a very cheap way. Emerging fields like metabolomics, proteomics, and precision medicines demand that there should be techniques which complement them, which help them. And one of the reason why metabolomics or proteomics has seen such a high jump in the last few years is due to the advancement in mass spectrometry, and also to address global challenges, which, as I previously mentioned, climate change, which is a very alarming situation for us, pandemics, very important, the COVID-19 pandemic, which happened just few years ago. We'll discuss how mass spectrometry and chromatography exactly helped in that. There are various industry growth drivers because of which we have so much focus these days on the growth of chromatography and mass spectrometry. The first is pharma and the biotech industry. This is mainly because these days we have new biological medicines known as the monoclonal antibodies, which need liquid chromatography and mass spectrometry for their identification. I remember when the COVID 19 pandemic struck, the first paper published said about alpaca. Alpaca is like a sheep kind of animal in Southern America, where alpaca was studied to have so strong antibodies against the COVID 19 antigens. And this was the first therapy which was introduced for COVID 19. The second is environmental monitoring. As I previously mentioned, polyfluoro aromatic system, which are now said as the forever chemicals. Once they get inside you, there is no way they are going out. They are in the pan, in the cooking, in the non-stick pan specifically you are using. With these needs GCMS testing to monitor them. The second is food safety. I'm sure you must have seen in the last few years, the food recalls has increased exponentially. Companies like Nestle and a lot are recalling their products. Why? Because there has been third party testing and it has come across that there is some things in the compound in the sample it should not be. For example, melamine. It has been found that milk in different states in India has melamine in it, which is harmful to the human body. This is one of the driving demand. And one of the very important is the forensics, in which new synthetic drugs, a lot coming from the fentanyl family analogs like the opiate drugs of recreational purpose, required advanced mass spectrometry approaches to quickly identify them. And this is what happens at the airport as well. If you have ever gone through an airport and see how they frisk people. So the first case study I'm going to present, which uses this analytical technique, is about food safety. Consider how LCMS transformed global food safety. Now, one automated run can identify hundreds of pesticides, sharply reducing health risks and expediting recalls. During the pandemic, MS-based diagnostic filled crucial gaps for COVID-19 testing, saving countless lives. So, chromatography and mass spectrometry was the first line of analytical or the scientific tool which was available in the market to help COVID 19. I remember I was in the UK during the pandemic, and we had a very big analytical lab, one of the best in the entire area. And I remember we loaned our gas chromatographers and liquid chromatography HPLC instruments to the NHS, which is the local health support system, and we loaned it to them for around 6 to 8 months because there was a very huge demand on their end. Then, in cutting edge hospitals, mass spectrometry is guiding the design of personalized therapies, ushering in a new era of medicine tailored to each individual's unique biology. Now the world of medicine has changed a lot. Now these days we can make tailored medicine specific for one person. I'm sure you must have seen the news of a millionaire who has spent around 21 million until now to reverse his age. All his medicines, all the supplements he consumes is all tailored to his body needs. And these days you can get a lot of supplements. They ask you a few questions and then they suggest you need vitamin K or D or so on. This is the real world power and promise of modern gas chromatography, liquid chromatography, and mass spectrometry. What are the future trends? I believe the first next frontier is about making analysis smarter and seamless. We already have technology which is a lot faster. Now imagine AI, artificial intelligence, which is a very hot topic these days, that designs your methods for you. Portable labs that deliver answers on site, and fully automated systems that run themselves. If you've ever been to an electrical lab, there are auto samplers. You just need to load your samples and it will do the entire job for you. It will load the sample, will do the analysis, will save the data, and will extract your sample back. It's all about elevating the scientist's role from operation to innovator and interpreter, empowered by technology that handles the routine. So it's freeing a lot of your time to focus on the analysis, on the interpretation of the data. Open access in mass spectrometry is already a very common topic in the Western world. They are considering making their analysis samples open access to labs throughout the world, which is it is also a very much debated topic as well. In the entire world, mastering GCLC-MS is a passport to exciting careers. Pharma, biotech, food safety, and government lab all needs expert who can operate, validate, and troubleshoot these advanced instruments. Global demand far outstrips supply. There is a major talent gap. Employers value those who combine technical powers with data analysis. Getting certified in GCLC-MS open doors to industry leadership, international mobility, and rapid advancement. In the past 2 decades, the demand for chromatographers and mass spectrometry expert has skyrocketed around the world. Be it abroad, be it India, the demand has increased exponentially, and so does the pay as well, which will Mr. Khaja talk about. Whether you aim for research, regulatory, or application science, these skills put you in high demand worldwide. With this, I conclude my session. I hope this was helpful. Thank you everyone for being a lovely audience, and I'll be more than happy to take any questions in the end. Over to you, Tanushree.
Yeah, I hope I'm audible.
Yeah, you are.
Yeah. Thank you so much. So, thank you so much, sir, for such an excellent and inspiring session, especially the examples and the practical aspects you shared were extremely interesting. Thank you so much. So, a quick reminder to all our Biotecnika and Rasayanika subscribers and all those who are attending this webinar today, please feel free to post your questions in the chat box. If you have any questions related to GCLCMS, please feel free to post your questions. Okay, it will be answered by our experts in the end. So now it's time for the second session. So I would like to invite our next speaker, Mr. Mohinuddin, sir. So over to you, sir.
Good evening, everyone. Welcome to this session, and thank you for the opportunity. First of all, and it's a very good insightful given by Dr. Sharma sir regarding the basics covered from the chromatography and liquid chromatography and mass spectroscopy. Yes, in daily life we used to deal with chromatography, um chromatography as well as mass spectroscopy and day to day analysis. My topic is about growth opportunities in allied chemistry. Today we will explore how we can build a successful career in the and exciting evolving field. We cover essential skills career paths and job roles and strategic growth in the field of electrical chemistry who are looking to advance their career and freshers as well as most of the persons as there. It will be a very good actionable insights. I will present my screen right now.
Sure, sir. Okay, so meantime, sir presents his screen. Just again, a quick reminder that you will be getting a free surprise gift. So please stay connected with us anytime. The link will be shared by our team on the chat box. So you just have to go through that
Apology
link, fill
for
the
my side.
form. Yeah, it's absolutely fine, sir.
Apology for my side. Yes, I'm audible and my screen is visible to all of you.
Yes, it's visible. Yes, sir, it's visible.
pathways to analytical chemistry, like Dr. Sharma told, strong educational background is a crucial, like B pharmacy or N pharmacy, relevant field is required, along with the proficiency in technical and analytical skills for doing routine laboratory work. Why it is? Because in day to day, the chromatography evolving. The earlier the technique is like it's very sophisticated right now. The advances in the chemistry and the instrumentations, everything evolving. The need of mass spectroscopy in the field of pharmaceutics, as well as identifying new drugs. Even small companies also, they are looking for the expertise in the field of chromatography and mass spectroscopy. They are looking for the candidates who's having a bachelor degree in chemistry, B pharmacy, and N pharmacy. Obviously, they are having very good chances of getting job into these relevant fields. And the next slide is key technical skills in radial chemistry. As I told, doctor told, proficiency in spectroscopy and chromatography. Why it is required, I will explain in detail. So if you want find any unknown compound from mixture of mixture of a samples, maybe it's maybe a drug, or it's maybe a forensic sample, or it's a environmental sample. What is my first idea coming to my mind is chromatography. The chromatography will separate mixture of components into individuals, and the identified and will quantify. And for unknown peaks, or if you want to quantify, or if you want to identify by what is exactly mass number, then spectroscopy, the mass spectroscopy will help. And after that, strong interpretation of mass spectral data is more essential to identify the compound. Example, this is a mass spectra. This mass spectra is recorded based on the M by Z and the relative abundance. This is the example I'm telling. Uh, the mass spectroscopy will works on the ionization of a compound. Like it's first it will ionizes molecular ions into daughter ions. So these ions will help to identify the molecule of unknown. If you have some unknown of molecule, we don't know what is exactly structure or anything. When you run chromatograph, you will separate the mixture, and when we connect that outlet into mass spectroscope for particular RT, you will get a mass spectral data. From this data, R&D scientist will predict the structure of that molecule. So this is happening exactly. What is that practical and way of correlating the data interpretation of chemical compounds, various samples. And essentially, of rapid technique for success is it's about synthetic chemistry or pharmaceutical science, like pharmacy, pharmaceutical science. So why they have exact knowledge on titrations as well as purification of APIs from the crude and extraction of API from the raw materials. These are the methodologies they will take care for getting 100% of high quality standards in the laboratory practices. And critical data analytical skills, ability to interpret complex data sets accurately means whatever the data they are getting, they are not relying on the they are relying on the analytical data itself. They have to understand, they have to interpret the data which is correct or not, applying their knowledge and skills to interpret the data based on their experience, based on the expertise. They will came to learn what type of compound is that was we are going to synthesize or we are going to identify. And the next slide is crucial analytical skills. This slide discuss about in analytical chemistry. In day by day, we will encounter with a number of problems related to the systems, related with the chromatograms, related with the data which are expected to come. Sometimes the data will not expected to come, like SA failures or SD failures. So we have to get a knowledge upon the instrumentation also, apart from theoretical and practical. We have to get instrumentation knowledge also. And the next thing is the problem solving skills required. If any of the problem is came, you do not stuck over there. You have to think out of box, move forward. Whatever I am getting the results is correct or not. You have to assess yourself. Then you have to apply your skills it will come based on your experience. Junior levels, you maybe very fresher or very new. When you go into that R&D or QC field, you can able to uh, get these kind of problems, repeated problems like RSG failures, instrument problem, SA failure, OOS, OODs. These are the terms. These are quality critical attributes of any analytical lab chromatography or analytical lab chemist, and we have to give attention to detail importance. Why? Because it helps to maintain a consistent result throughout the experiments. When you develop a method, the method should be a robust in the nature. We have to keep in that mind. We have during the development, whatever the method we are developing, it should be repeated, or it should not have any problems at the plant level also. It's like a detailing of your method. Once you develop method, it's not your job done, and until or unless it gets success at the plant level. That is what my intent to explain you. Most of the people are like you are freshers or B pharmacy or M pharmacy freshers. So when you go into the field, you can able to feel what I'm feeling right now. Earlier, I was like a electrical average guy. I just scored around 70 30%. Then I got a job in the electrical R&D in the pharma, and then I used to learn the techniques one by one and evolving, learning the techniques to real time. How the cutting edge technologies are evolved. Earlier of 2012, the techniques is not there right now. They are very much advanced. I'm telling you, they are very much advanced. Even though AI also integrated into our analytical chemistry, it's giving a very wonderful output. And coming to the next slide, the career progression in analytical chemistry. So this is a mostly the very important section. Most of the people will don't know what is the roles and responsibilities in the different organization. I will explain in a simple way. This is like a this is not this is our company profile. It will depends upon the company to company. It will changes from one company to another company. The basic role will be like lab technician, maybe or junior research associate, with whatever they call as. This is an entry level role with hands-on experience. That's it. You need to don't need to have any expertise. We have to only a theoretical knowledge about what is a chromatography and what is a mass spectroscopy and how it is works, and what is a theoretical background and IR, NMR, XRD. This is theoretical background is enough for a technician. It's not in expertise matter. Then if you are getting a more experience, you are promoted to higher roles like research associate. The role is you know collaborating in the research projects and taking end-to-end responsibilities, interpretation of data, whatever the outcomes are, whatever the requirements of the analytical methods. But these are the things will comes under. And when you are getting more experience, you are pushing to the a senior role like senior scientist. In the senior scientist, the leadership skills, like if you are exposing more number of problematic or like motivating team, and giving a planning, execution, planned execution. So these are all comes under senior chemist. Based on the experience, it will drives into one lab technician to senior scientist. The role is a leadership role with advanced expertise, providing solution to the problems online and guiding to the juniors, and taking care of everything, and what are the organization requirements and where we are lacking, what need to do if anything goes into the plant level, what we have to take care, what are the problems will comes if you take these decisions. So these are main key aspects coming to the senior scientist level. And the lab manager level, we know we are getting more experience, we are promoted to the higher level and higher responsibilities, and it's a, like a overall management kind of thing. So you already have the from starting to ending, the lab manager having all exposure to the all the techniques and all the relevant fields, and he is into the management. He will management the project, what are the cost of the project, and what are the cost cutting have to take, and what is the organization requirements, and what the competitor is launching a product into market, how much we can reduce the cost, what are the, and then also which instrument has to take in coming to the problem, like how much which column he has to take, like lesser cheaper or more costly. So these are the decision taken by the lab manager and the director level. It's a very top level. It's like a taking of strategic decisions, which projects needs to be taken in the pipeline. They are dealing with the regulatory team, IPM, intellectual property rights. So, they are group of peoples. They work together and they deliver in a pharma or in any kind of fields. In some other government organization also there, they have the same roles, but they have the same designation, but the responsibilities different. Like you can go. This is I'm telling you, but pharma. If you move to the bio research lab, the same chromatography will come. And if you go, if you can take example as uh, another science, bio, same kind of designations will come to you. And the next listen is a very important thing I need to discuss about. So salary trends in analytical chemistry roles. I want to add one more thing here. So it's like a example or overview. It is not fixed. It maybe depends upon the company to company and tire, like whatever what I mentioned, tire city of the company. Example, Bangalore, we have some different packages. In Bombay, they have different packages. And Hyderabad, this is a overview or generalized statement I'm given. It's I've taken multiple questions from my juniors and other what they're getting paid, and I mentioned these ones like junior research associate they're getting 3.5 lakh per annum, and if you move research higher roles like head of the department, you get 35 lakhs per annum. Sometimes they will get more depending upon the roles and everything. As you know, the demanding of the expertise is always pays. So if we have more expertise in one department or any multiple department, you have exposure more. So you have the getting of your resume, or you are getting picking of you in a job is very easy, very easy in this computer environment. Like a salary is like a is don't depend upon the salary, like junior research at 3.5 lakh per annum, but it is a just example I'm telling. Don't stick it, this is a 3.5 lakh I'm getting this very much or less. Don't demoralize. I'm just giving an example. What are the salary trends in the electrical chemistry and role? Sometimes it may be higher to the, like, what is innovator companies, they will pay more. The basic of the salary of around 5 to 6 lakhs. If you are in IIT background, they will pay you more. Sometimes 10 lakhs per annum also they will pay. So these are the process also there. I don't stick into that 3.5 5 lakhs, 6.5. This is for understanding purpose. I have kept here. Uh, Coming to the next slide. Uh, so who's hiring? So mostly leaders leading research labs like our India having mostly generic companies, Dr. Reddy's, Arvindo Pharma, Teva, Cipla. So these are the leading research lab. Forgot to mention some of the names, some of the companies' name. But these are the leading research lab. They are currently working on the innovative medicines of delivering different kinds of medicines to cheap and affordable medicines. Like our doctor is what we are trying to do is we are bringing cost down to the innovator version, the generic version with less cost effective, to affordable uh, to the normal humans, like the normal humans for regular use in terms of chemotherapy. And the pharmaceutical companies, major pharmaceutical companies you already known, like drug development. We are India is more focusing right now. The even smaller startups also doing, putting their hard money into that drug delivery research by using artificial intelligence. And some government agencies like forensic science, Recently, a government forensic science, the Delhi they have given a employment for the open hiring for the candidate who having experience in the field of forensics. So forensics, one of my colleague got selected in that. Oh, so these are the not only biochemistry or chemistry, M pharmacy or B pharmacy, forensic science also it comes under your research field, to getting a job in a government agency. Also, don't fix that chemistry only giving research job. Don't think that in our department we have non-technical persons like from the pharmaceutics also there, and from forensic science also there, and BSc background also. So it's all about their interest. If you are interested to learn, nobody will stop you. The companies are there. They will see you. They will see only your potential. How you handling, how you answering the question, and what you know, how you will help them to achieve their goals. That is a simple and basic funda. I'm telling you. If you are giving an interview, just match their expectations, and then you will crack the interview definitely. And coming to certifications, I need to tell you, in LinkedIn, how many of you using LinkedIn, I don't know, but I used to take Six Sigma certificates. It's like a one step ahead of my practical and theoretical knowledge. It will add on skills. These skills will help you in the future. So certification is essential. Certification is an identity that you have mastered your skills into that. And keep searching in LinkedIn. You get like if you're like webinars, if you are getting, if you want to learn any like GC or chromatography, like I need to this site is there, Agile chromatography. In this webinar section is there. You can go and search webinars for the relevant topics. You can find GC webinars and column. So uh, so we can enroll GC and MCA webinars if you want interest. Uh, these are all at recorded sections. Um you can select, and we can whatever relevant you can want expertise into. If you want to explore more into GCLC, you can go through Agilent sites. Every sites have their own uh, apology for that. Every site has their own webinars, so you can go and take the lesson from and learn from that, learn from those. An internship for practice experience, our Dr. is open to for all young you guys, and keep posting in LinkedIn for in internship, and keep tagging Dr. Reddy's. You will get a practical experience, and they sometimes they will pay you also. For us, we don't have that opportunity. You guys are very lucky, and Dr. Reddy's, Deva, and other all the Cipla, and all the companies are giving you paid internship. Also, keep looking in the LinkedIn page. Go to that company website, careers, and then post your resume and approaching them, and it will comes under it to networking of professionals, and professional development strategy. It will comes under that. Join a professional organizations for research and connections. Like once you get into an internship, and you might have interacted, you might have chance to interact the experienced guys, then how they are thinking, how they are getting into that position, how they are approaching to a problem, how they are doing method development and method validation, what is a theory and what is a practice they are following. And then once you get into the organization, you will get, for that you need your practical and theoretical knowledge. That's it. No one will stop you. Once you get a theoretical and practical knowledge, but a chemistry or analytical chemistry, a B pharmacy and pharmacy, just get a practical knowledge. That's it. No one will stop you. uh, Crafting your resume for chemistry, most of the things, what I do is, I use Canva for my resume, for resume making. I use AI, Canva AI. So these, I just put my expertise and my requirements into Canva AI, and it will generate my resume. And please use AI Canva for giving more presentations or giving more relevant designing templates of your resume. It will very helpful to you. It will not take less than 5 minutes or 10 minutes. Uh, the AI, artificial intelligence, is a machine. It will work for you. Uh, it will work for you and on your commands, and it will generate a PPT or whatever you want, like resume or anything. Just you give your skills, your experience, and which organization you're going to applying, and just you give small information. It will generate end-to-end details, and just you enter your details of photograph. That's it. That will help you. It will reduce your time, as well as it will open your another dimensions. Don't relying on others senior resume or anything. Just go and look for what is organization asking, what are the job responsibility. Please copy paste into those job and responsibility into your resume. It will having a more chances to pick your resume. You pick your resume. It will give more number of chances that I can tip. I can help you to find out pick your resume. And I told you earlier, networking and professional management. I used to do. I generally mostly applied jobs in LinkedIn, Naukri, Times, and Shine.com. So these platforms will help you to, as a fresher, don't look for salary. Just upload your resume and get interview calls regularly, because the job market is open for us, because the billions of marketing of generic is expanding year to year. You can find one website, LCGC. In that you can search for salary trends in electrical chemistry. You can find a PDF. What are the current trends, what are the current job trends, what are the salaries, and what are the technical skills they are required. Networking and professional management will help you to get a desired job, like desired job into the organization. And thank you so much for your patience, and thank you for giving the opportunity. If you have any doubts, please reach out me, my mail ID, and my LinkedIn profile. If you have any doubts, it's over to you, ma'am.
Thank you. Thank you so much, sir, for such actionable insights about career growth and industry expectations. Your session was highly informative. Thank you so much. So now we will open the floor for the questions. So I request all the attendees, Biotecnika and Rasayanika, on our Biotecnika and Rasayanika platforms, all those who are watching this webinar, attending this webinar, I request everyone to please post your queries in the chat box, and we will be happy to take them one by one. So first question I will be taking of Sudeep Day. Sudeep Day wants to know that is it possible to identify a totally unknown material along with its chemical formula by GCMS? Also, similar question is from Mumpy Roy that what about separation of unknown sample mixture? How this will be helpful for specific identification of components. So basically, our attendees wants to know, sir, that whether GCLCMS technique can be used for separation of unknown sample mixture, and also can we identify the components in that mixture. So I request Dr. Anirudh, would you like to answer this question?
Sure, sure. Thank you, Tanushree, and a very, very interesting question, Sudeep. I think analytical chemistry takes too much pride that there has been so much technological advances in the past 4 or 5 decades. But unfortunately, you there is no technique which can completely identify a something which is completely unknown. It will take complement of spectroscopy, spectrometry, IR analysis, and so on to identify something which is completely unknown. This is how they identify samples of extraterrestrial asteroids, Mars, Moon. That is how they do it. They use a component, they use a complement, all the analytical technique, but not only GCS is sufficient enough. We complement other techniques like spectroscopy and so on, and it works so marvelously and so beautifully. So yes, it is possible by the complementation of techniques.
I hope this answers your question, Sudeep and Mampi Roy. We have another question from Mruduvan Rebelli. Uh, so the question is how to pursue PhD in analytical chemistry as a young researcher in India or abroad? What are the opportunities? So, Mr. Mohinuddin, would you like to answer this question, sir?
Um, yes, there are like paid paid universities also there, and also in university they will sanction some free grants for you. I would recommend Kaust University is there. They will give you like they are exploring right now environmental chemistry. They are open for that, like enthusiastic chemistry or a B pharmacy or M pharmacy, any educational chemistry background, they can apply for that. It's like a free of cost. We don't need to pay any tuition fee over there. You can once you got selected, there are 3 to 4 interviews you have to give. One is technical as well as HR, like how you formal interview. There are 3 to 4 rounds. Once you clear that interviews and you got selected for that one, they will pay you annually, and you will get a medical composition as for you and as family. They are some university also there. You have to pay and you have to learn. In India, I would say IITs are best for you, because once you get to the IIT and you came to know what is exactly research means. I would say Sharma sir already having that experience, he will explain it better. Exactly, research will start in IITs or NIT only. I'm not telling you smaller colleges or universities have, but they don't have that much capacity to explore your knowledge. If you want more mass exactly, you want to do mass spectroscopy or any or NMR, you have to rely on the other third party labs for your sample to get analysis done. So in NIT, IITs, you have that facility and you can explore your PhD thesis without any interpretation. That's what I'm going to convey her. India and abroad are having equal opportunity. Whether if you want to apply in India, you go for IIT, NIT, whichever relevant, suitable for you. And if you want to go abroad, Saudi, Middle East, Scout University is there, and rest of the world, US, UK, there are mostly number of university are there. Find a PhD.com, you can Google it and you can search it. Based on that PhD score, then you can select the PhD university. I hope I answered the question.
Yeah, yes, definitely, sir. Dr. Anirudh, would you like to add something?
I totally resonate what Mr. Khaja said. India also there is so many opportunities, but we do have a big number of opportunities abroad because analytical techniques need investment, mass spectrometers and things like that. These are not cheap. It does take a good amount of capital to make a analytical lab. So definitely, Chaos University, it's very good. They have got the recent uh, analytical lab. I have seen their website, and it's very good. There are many other universities like in England, in the US, and again, it doesn't cost you anything. You should have a strong profile, and you can definitely get through a PhD.
Thank you, Mr. Mohinuddin, sir, and Dr. Anirudh, sir. I hope this answers your question, Mriduvan. So moving over to another question, we do have some technical questions, sir, based on the working of GCLCMS. So first question is, what are the major challenges in GCLCMS chromatography? This question is from Rajan, sorry. So anyone wants to take up this question, sir? Anyone?
I would like to take that question.
Yes, sir, please proceed.
Uh, could you repeat once again? Apology for that.
The question is, what are the major challenges in GCLCMS chromatography? Or I think the Rajan wants to know what are the major challenges in GCLCMS?
The major challenges, you are clubbing the two different techniques. First of all, GC and LC are comes to in the chromatography. They works on different principle and different purposes. See, like Dr. Sharma told, the liquid chromatography works on partition, interaction of compounds of interest. They are moving in the mobile phase, with the stationary phase, and the separation will happen. Okay. In the gas chromatography, first the sample it get compressed, and then it will separate onto a column, and then it later part is detection. Any chromatography, there are two things. One is sample preparation, injection, separation, and detection. So, in preparation, there are number of challenges depending upon the type of molecule you are going to analyze. Suppose, example, bio samples you are doing, you need to extract plasma, all other cell mass, and your compound of interest into one side. It will be happen doing a filtration technique. Simply different technique. It's sample preparations. You need to prepare sample before you going to inject into your chromatograph, whether it is liquid or gas chromatograph. Like if you want analyze residual solvents or you want to measure ethanol content in your blood sample exactly, then in this case HPLC will won't work, gas chromatography will work. You have to prepare a sample in a headspace file, and you have to prepare some mg per ml concentration, and then you inject and you will get a data. It's a coming to biology. And the pharmaceutical compounds, the main thing is solubility. Most of the compound will soluble in water, it's no issue. And if something is soluble also, they will not separate. It's another separation. We have to change the mobile phase colors. Different kind of approach. And for GC and LC, for GC and HPLC, the sample preparation is mostly a critical step in the development part as well as the QC part. Most of the problems will come from the sample preparation only, because instrumentation will give you like RSG failures or something like your data is not acquired due to some instrument problem breakdown. These will come from instrument side. So from mostly method side, the sample preparation 1% to 1% understanding is different. Well, that same written statement, it can be understood by different ways in the same laboratory. Given one statement, the sample should be weighed in RH. The person should ask what is the RH and what is the limit. So these are the technicality terms will comes to there. I will summarize the question. The sample preparation techniques depend will be different for the pharmaceutics, as well as environmental sample, and the liquid chromatography sample preparation is different, and GC sample preparation because we can't inject water directly into GC. I hope your question I maybe answer. If you want to add more, Dr. Sharma, you can add more to it. They can get a more clarity on that.
No, no, sir. I think you did complete justice to you. You did it much better for me. So there isn't for me to add.
Yeah, I
Thank you so
completely
much for
agree.
kind words.
Yes, I completely agree for such a detailed explanation. Thank you, sir. Definitely, I think Rajan, it's this answers your question, right? So now moving over to a few more questions, sir. So I can see many attendees, they want to know the app, like what kind of sample they can use if they are using GCLCMS. Like we have a question from consistency, that will we be able to extract and purify microbial metabolites using GCLCMS, and do we need known concentration of those metabolites to identify them? Similarly, we have a question from Venkateshwar Reddy, and so he wants to know that in toxicology, can we use GCLCMS? So I just summarize that whether we can use GCLCMS for extracting and purifying microbial metabolites, or can be used in toxicology, and if you can provide some insights on this, and what are other fields we can use GCLCMS, like proteomics or metabolomics, can it be used? Dr. Anirudh, would you like to take up this question?
Sure. So would you mind in toxicology how and where we can use? So a lot of, as I said in my slides, that a lot of medicines that we prepare these days, they have certain sort of impurity, and there are permissible limits for that. For example, FDA says in paracetamol the impurities can be up to the ppm level. Some medicines, those who are more complex in their pharmacokinetics and the pharmacology, their impurities application level in that state is just ppb. So, and it, you will be surprised to know that biotechnologists, those who have a biology background, use more GCLC and MS more than a chemist, because the techniques we have available in GC, for example, the electrospray ionization, which works very well with large molecules, protein molecules. So it can be definitely applied in the toxicology and even other fields as well. And like to reiterate again, if you think you are a chemist, you use less GCLC and MS more than compared to a biotechnologist. Students with a biotechnology background need more GC to characterize their protein and to even purify it. So I think this answers the question.
Yes, yes, definitely, sir. Thank you uh, for the detailed explanation. So we have one very interesting question from K Khan, that what are the benefits of GCLCMS in diagnostic lab, and can one can get job for this role in diagnostic lab?
Like, related to the question, recently, like, if you want to analyze like hormones in your bloodstream, the traditional technique is there, but it won't give a reliable results. If you want more particular about the results, yes, you can go for LCMS methods also there for identification of like testosterone and progesterone hormones. Yes, we can do analyze the samples by GC. And I want to add that previous question to some more points. The basic the thing is, when you go for HPLC and when you go for GCs, the compound is UV active. When you go for HPLC, when your compound is volatile and thermally liable, then you will go for GC. The basic criteria for selection of a technique, and the sample preparation technique is different, and separation mechanism it will be followed. Yes, you can. Yes, you can get job, but many companies, like how many data center they are following these past practices, we don't know. But yes, they are using uh, for drug and trace analysis, and for sometimes like third party analysis we used to give for what do you call bio studies. We used to give sample bio studies. Some companies are doing. Yes, if you get a chance to learn, yes, you can explore the techniques.
Thank you so much, sir. Moving over to one last question. So, everyone, I'm really sorry. We are just like, because of the time constraint, it won't be possible for me to take up all the questions. So, I will be taking last question. So, there are a few questions which is related to like whether the student is in final year. If somebody is in final year, so there is a question from Jeep, that is it necessary for MSc Biotech final year student to learn the use of GCMS before going over, before moving ahead for any internship or any research work or any project? uh, Maybe many students might be having this question, sir. Maybe they are not from the chemistry background, so if they are from biotech background or microbiology background, or if somebody wants to do some research work related to proteins or secondary metabolites, as one of the attendees has mentioned, or maybe related to analytical chemistry. So how important this GCLCMS technique is, if anybody wants to pursue research in any of the life science field or analytical chemistry field?
So I would again reiterate the same thing. Biologists do a lot more mass spectrometry and chromatography than the chemist. Chemist has a lot of other techniques, spectroscopy and electrical techniques, UV vis, and a lot other things that we can use that does the job, NMR a lot. So, but in biology, we do have a limitation where we use these techniques. For example, as I said, electrospray, it's such a gentle ionization process. It leads to native identification of your proteins. So if you're doing MSc in biotech, I would say chromatography and mass spectrometry is absolutely indispensable for you. As this the poster for this session said, 70% entry level jobs need GC, LC, and MS as a prerequisite, which I think Mr. Haja will agree with me. So whether you are biotech or somewhere near around that, even microbiology or in chemistry definitely, or even in biology anywhere, chromatography and mass spectrometry are your friends, which will go a very long way with you. They'll be with you if you are in this field for your entire life. They will be with you. So, yes, to answer your question, it's very necessary, important, and required for you to have these skills before you open yourself to the job market. I hope this answered your questions, Jay.
Definitely, definitely, sir. And I can see so many questions out there, but because of the time constraint, I won't be able to take them. So those whose questions are not taken, please don't feel disheartened. You can post your questions at cst@biotechnica.org, and we'll be happy to answer it there. So everybody, it's time for the surprise gift. Our team will be sharing a link in the chat box. So you can fill out you can click on that link, and there is a form. You have to fill that form, and you can receive your free gift, which is an ebook on mastering GC and LCMS, a career handbook. It's a complete guide for life science, chemistry, and pharma professionals looking to excel in gas chromatography, mass spectrometry, and liquid chromatography, mass spectrometry. Also, I'm very much excited to announce our upcoming GCLC MS course, which is starting from 22nd September. I can see, Sushri, you have mentioned that you want to gain experience on GCLC MS. So, and many more, I'm sure, after attending this webinar, you get a clarity that how GCLC MS is very important. It doesn't matter whether you are from biotech field or microbiology, or and definitely chemistry. So, this particular technique is very, very important. And to uh, and realizing the importance of this technique, we are coming up this particular course, which is starting from 22nd September, and we look forward to welcome you and help you to take your career to the next level. So with this, we have come to the end of this very insightful webinar. I would like to thank our experts, Dr. Anirudh sir and Mr. Mohinuddin sir, for sharing their deep insights, practical tips related to GCLCMS that highlights the critical role of this technique in research and industry. Also, I would like to thank all our Biotecnika and Rasayanika subscribers, students, and all the attendees, those who have joined this webinar today. Thank you for joining this webinar today and actively participating in this knowledge-packed webinar. We hope you found the session insightful, this webinar insightful and motivating to further build your skills and advance your career in analytical sciences. So thank you and take care.
Take care.
Thank you, everyone.